Okay, why don't we get started? Thanks everyone for coming on the day before the long weekend. Uh, you guys are the real troopers, apparently. um, so today I'm going to be talking to you about approaches to functional activity mapping during natural viewing. And I'm going to mostly focus on natural viewing, but this is more about using naturalistic stimuli than just simply visual ones. Because the idea here is that you can use more naturalistic stimuli to try and probe how the brain responds in the real world as opposed to during very controlled settings. And it doesn't just have to be visual information. You can use auditory and possibly tactile in some ways if you can come up with an experiment that would work with that. This is more about kind of the design choices that go into that, and then how do you deal with the loss of control that these choices may give you. So first, I'm going to get a little bit into kind of the visual experience that you experience throughout the world. Those design choices, how we can go about figuring out how the brain is responding to these things, and then some other kind of experimental tasks that can be used using these similar types of tasks, looking at kind of how individuals respond similarly or different from each other, or even to each other. So visual experience. So the brain has to take in all this very complex visual information, and your brain is excellent at this. It takes in vast amount of information constantly through the eye. You see this picture and you immediately recognize it for being a set of boats, some people walking, a couple of people here, there's people in the water back here, trees, but all these things are just little patches of paint on a painting, yet your brain is able to quickly interpret it as what the artist meant for you to see. This all happens through a very complex visual system that we have. The stimulus comes in through the retina, goes down through the brain, goes through the brainstem, and then comes back up into the visual cortex, and then the visual cortex spends a lot of time processing this, and then actually sends it forward back through the brain to the temporal lobes and prefrontal cortex before making a decision on what you just saw. So your whole brain is involved in the task of just even interpreting this painting. And one of the great things about fMRI is it gives you a chance to investigate the whole brain. But all these things are happening on a very fast time scale. And so oftentimes what people do is they bring everything down into a smaller space so that it's a little bit more manageable to deal with. But if you're going to use a naturalistic stimuli, you have to account for the fact that there's tons of information coming in all at once, and you can't just downsample it to one particular feature, say a grading. So the real world looks something more like this in that painting. There's lots of motion, there's different actors. You have to pay attention to what each individual is doing. And all of this is going on along with background information. Your brain's able to process all of that at once, even though your fovea is only picking up a small portion of that at any given time. For instance, your eyes, this is a bunch of different trials of a monkey watching the same movie, and you can see on individual trials and even at different time points during those trials based on the colors, their eyes are all over the place. They're constantly saccading around, changing the visual input that's hitting the visual cortex. And so there's a ton of complexity between both the stimulus and your own movements through the world that have to be accounted for when the brain processes just the world around you. This is the same with auditory stimuli. Auditory stimuli can be particularly interesting because you really only get information from audition through temporal processes. You need a stream of frequencies to actually hear words. There's no individual, you can't put up a snapshot of a word. There is no snapshot of a word. You have to have time to be able to do these things. And so when trying to do experiments, we have to take all of these things into account. So there's a lot of different trade-offs that neuroscientists and psychologists have to make when they're planning an experiment, sometimes without even really thinking about what they're doing. They're trying to maximize the trade-offs that are involved in choosing stimuli and experimental designs. So if we think of an experiment as a continuum that can fall anywhere from a very artificial but controlled stimulus to the real world or a natural stimulus that's completely uncontrolled, you really have the whole space to work in. And wherever you put your experiment, you're going to end up with certain types of trade-offs. So if you have a very controlled stimulus, this is where a lot of neuroscience started, where you take a anesthetized animal and you present a grating in a particular portion of the visual field, you're going to get very strong control over what's going on in the world. You've minimized as many variables as possible, but you're also as far away from the real world as you can get. So the question then has to become, how can that be translated into what's going on during natural vision? How translatable is it? How much does it connect? So these advantages and disadvantages to doing this kind of artificial stimulus. On the far end, we can imagine an experiment where we have an animal out in the real world. It has a set of electrodes in its head, and it's navigating its own environment with hundreds of other monkeys around, and through telemetry, you're recording what the animal is, what's going on in the brain. Now, this is going to tell you exactly what that area is doing during the real world, but the problem is it's going to be completely uninterpretable, at least at our current level of understanding the brain. My PI, David Leopold, likes to joke that if you do something like this, you'll probably be able to tell after hundreds of hours of recording whether the monkey was awake or asleep, and that's about all you'll be able to tell from this data currently. But those aren't the only two extremes. We can fall all along this continuum. Most people, when running fMRI studies, sit somewhere closer to the artificial controlled, but they get some more real-world type stimulus in there. A good example of that is a lot of people use face stimuli to probe social decision-making or just object processing, these kinds of things. So with a face, you have a particular stimulus. We're going to show it for a couple different times. Most people do this in a block design. They're going to present a bunch of faces, then they're going to present some scrambled images or objects or something like that, and then they're going to do a contrast between the fMRI during the viewing of a face versus the viewing of some other stimulus set. And this has worked great for modeling a lot of different processes in the brain. In the rhesus, you can find things like face patches or color patches. In humans, we've found the FFA and PPA, places that respond to faces versus places. And lots of other types of experiments have used these types of designs. However, we can start moving more towards recently people have started to move more towards a more naturalistic design setting, where they're starting to get a little bit closer, maybe not as far over as that dot says, but a little bit closer to nature, the natural world. So in this case, it's something like this: a monkey or a human watching a set of videos and then trying to understand how the brain is taking in all this complex information at once and processing it. The trade-off here is you've probably allowed the animal to start looking around more, so you're not asking them to fixate on a particular topic because you're asking for natural behavior. So now you have the increased variability from eye movements. And you also have the fact that there's a lot of information that's impinging on the brain, and you might not be able to tease out exactly which piece of the frame is driving a response in a particular area because you haven't isolated each individual variable in this case. So a lot of the goals of the studies that do this is to try to figure out ways to either incorporate this variability into the design so that it's a useful tool, or constrain it still, maybe post hoc or using some analyses to actually be able to pull out which particular feature of a video is driving the brain in different ways. And people have done this a few different ways, usually converging on a similar type of thing, which I'll get to, usually using some form of like a regression analysis. So the classic example of these studies is a group of people, starting with Uri Hassan at Princeton, presented a bunch of human subjects with the good, the bad, and the ugly. Here's just one of the classic scenes from it. So the subjects were just required to lay in the scanner and watch the movie. And then afterwards, it was on the experimenters to actually be able to try to pull out what actually happened in the brains during that time. And so a lot of experiments in humans, for better or worse, have actually used the good and the bad and the ugly as their kind of like go-to visual stimulus. And this, I have a slide later on that gets a little bit at this, but this is a good and a bad thing. One, it allows groups across different universities to compare their results to something because they've used the same stimulus. But on the other hand, there may be special things about this particular movie, the way it's cut, the color scheme in it, that if you don't start using other stimuli, you don't look at other things, you're going to, again, get in this trap of using a controlled one particular type of stimulus, and you won't get the full breadth of what's going on with these naturalistic stimuli. So one of the ways that researchers have gone about analyzing this data, the collection of it, if I haven't already stated this, the collection of this type of data is extremely easy. It is literally put a subject in a scanner and ask them to watch a video. I've done this. I'm going to start talking about some of the work we've done with the macaques. It's essentially like going to the movies for them. They sit in the scanner, they get juice for watching a movie, and they'll do it all day long. With the humans, it's kind of very similar because it's much easier than a task where they have to stare at a dot and watch a grading go by. Good, bad, and the ugly is far more interesting than that. So from a data collection standpoint, the data collection's very easy. It's the analysis that gets particularly hard with these kinds of experiments. You have to figure out kind of from scratch where to go. So one way that people have done this is to do something like feature coding. So what this involves is using algorithms or humans to code all the relevant features that you can think of in the movie and then try to regress those feature time courses against the actual responses in the brain. So some of the easier things to do are to get low-level features that people often attribute to responses in early visual cortex, such as capturing the luminance or motion in a frame, the spatial frequencies at different scales, or the contrast. Any other low-level features that you can think of, you can often use algorithms to pull these out of the movies, making these less labor-intensive. You can run them through some form of a script. However, there's much more difficult ones to code currently, because algorithms don't do a very good job with these, are things like high-level visual features, such as the number of conspecifics in a scene, or the number of mammals or humans. And these things, what becomes difficult here is these things overlap with each other, right? So if a monkey is watching these movies of other monkeys, but they also have things like occasionally they have polar bears or a deer, stuff like that. Some of those things are conspecifics. The conspecifics are all also mammals, as well as those other features. So there's overlap between the features. So something that happens a lot with these stimuli is that you don't have clearly orthogonal features throughout the movie, which is something that you usually try to set up with in your block design or event-related design, where when you manipulate a particular feature, it's going to not co-vary with another one. And so there's often covariance between these features, which is something that you have to deal with when doing the analysis. You can even go as high as to try to understand social behaviors, such as different behaviors that the animal may be doing or the humans may be doing. So you can code for the aggression, grooming, feeding, play in the videos if you're using animals. One of the papers I'm going to talk about in a moment does similar things with semantic labels like driving and stuff like that, like motion, like moving through a scene, running, that kind of stuff. And they look at different kinds of semantic labels. So what you end up with after spending quite a while coding these movies in various different ways is you end up with a time course for each feature. And those features are going to be somewhat correlated with each other, but what you're going to try to do is figure out ways to decorrelate them with each other and look at the how they are influencing responses throughout the brain. So the simplest way to do that is that you basically can take a given feature and correlate it with activity throughout the whole brain. So here we have the feature of one face throughout one of the movies that I presented the animals. And here is a voxel from an area that's known to be within a face patch in the animal. And you can see that even in this very highly variable movie, when this voxel is coding for the presence and absence of faces. So when there's a face on the screen, the response goes up. When there's no face on the screen, the response goes down. And so you can do that across the whole brain. This is moving on to a study from Jack Gallant's lab, where they did a similar type of analysis, not the exact same one that we ran, but a very similar one, where they presented a whole bunch of movies to their subjects. They had the movies labeled based on a whole bunch of semantic categories. And they had a little tick for when each category was present or absent in the frame. So they had 1,705 nouns and verbs. They also obviously have the fMRI activity from the responses to the movies. And so what they then do is, through a particular regression model, they can calculate the weights that for each of the categories on a given voxel. And they can get a model that represents for each voxel the various weights for all of those verbs and nouns. And what they can see from mapping out the weights of a particular voxel, so this one comes from the parahippocampal place area, that it has a larger response when all these more structural type of things like, structure, a building, shop, room, vehicle, all these like artificial things are on the scene, and it has a reduced response to things that are actually animate, like mammals and birds and communication and talking, which are going to involve animate objects. So using this method, they were able to pull out at the individual voxel level kind of the coding of that voxel in a more real-world type of setting. And now for their analysis, they see that in the fusiform face area, in the parahippocampal place area, they're able to get similar types of maps that you would expect from when you did block design. So now the one person you might ask, well then what's the use of doing this if a block design is much simpler to run and analyze? But what happens is it's not just that it reproduces these things, you actually get a lot more information on top of that as well. So in addition to just reproducing these more simplistic results, they also are able to look at how when you run a principal components analysis on the feature space, you can see that there's different types of features are loading on different principal components. So the first one's really heavily loading on whether or not something is alive or not. The second one is loading on social versus non-social type of things. And I'm trying to remember what they called the third and fourth ones. I don't at the current remember. But what then they can do is plot across the brain how the loading is on all of these different features. And what they see is that there's this smoothly transitioning set of feature space, semantic feature space across the visual cortex. So this is something that wasn't really seen before, showing that that across features, there's a smooth transition from, say, like early visual cortex into, let's see, try and draw it out. So see, these early visual cortical areas are doing more things like color, loading on one particular set of features. Then as they move anterior in the brain, it starts to slowly change towards yellow on the opposite side of the sphere. So it's now coding for different material, but then it actually smoothly transitions back into the other and then back again. So there's these groupings of voxels that are all coding for various different types of features, and they're kind of smoothly changing along the cortex. So we saw, oh, sorry. One other thing, I don't have a slide up here for it because it's very similar, but they recently published a new paper in 2015, I think, maybe it was 2016, that showed, 2016, that showed that you can do this as well with auditory stimuli. So they did a very similar type of analysis, having the subjects listen to stories, NPR stories, and saw that you can map the semantic word space across the cortex. And what was interesting about that paper was that they don't just get auditory areas or motor regions, but that a significant portion of the brain is actually responding to these only auditory stimuli. And that includes areas like early visual cortex and areas that are known to be very visual in nature. So it suggests that in a more real world setting again, that the whole brain kind of gets involved in processing these stimuli. So what we did is a very similar type of analysis. We presented these monkeys with a whole bunch of set of movies, and what this shows here is the cumulative variance explained across the movies, and then we can look at individual areas across the brain and try to understand how they are responding to the different features throughout the movie. So here I'm going to show you some what are classically known as visual regions, early visual regions, and what we see is that motion is what's driving area MT. This is to be expected because it's known to be a motion area, but really almost nothing else is accounting for the variance in this cortex. When we go to early visual cortex, such as V1, we see that the majority of the stimulus is again motion and but contrast as well. V4 is also being driven by motion. And so these areas we'd expect to be driven by motion and luminance. I don't have foveal V1 up here, but foveal V1 is interesting in that it's driven more by luminance and contrast than motion. It's one of the few areas that is in the early visual cortex. What becomes more interesting is when we start looking at areas that are classically known to respond to objects. So these are labels for the face patches. The labeling doesn't matter so much, but this is the middle one, the posterior one, and the anterior one. And when we look at those same areas, what we can see is that faces in green here are activating these areas, as I showed you earlier, but what's really driving these areas is the motion in the scenes. And as I was telling you earlier, you have to account for the shared variance in these things. One that might wonder, well, faces move, so is that what's driving these areas? Well, we actually can account for that by, we used, in our particular analysis, we used a stepwise regression, which subtracts out the variance explained by one given feature before applying another feature. And so additionally, motion and faces were not correlated with each other. So they were actually fairly orthogonal in our particular movies. This wasn't designed, it just happened to work out that way. But so between having orthogonal variables and the stepwise regression, we can show that these areas are, that the faces are, it's not just that the faces are moving that's driving this, it's that the whole scene is moving, that's driving these areas. And then on top of that, there's or underneath that, there's a face response. It only modulates the area a little bit compared to the actual motion in the scene. Another way to look at that, we can look at the ventral visual stream. We can break it down by what's in the ventral bank of the STS versus the lateral surface of IT. These are two known streams along the visual pathway that have been both implicated in object processing, though the ventral bank and the STS has also been implicated more in motion than the lateral bank. However, what we see is that while the ventral bank is more activated by motion, the driving feature in both the ventral bank and the lateral surface of IT is the motion in the scene. So what this suggests is that these areas are really driven by these more naturalistic stimuli, because the motion is simply the difference between two frames, which is basically what the real world is, something constantly moving and changing throughout. And so this suggests to me that taking into account these more naturalistic stimuli and the responses to them is going to become increasingly more critical in understanding how the brain's actually processing stimuli in these regions. So one thing that I've kind of left to the side, but I mentioned earlier, is that there's not just stimulus motion. The whole time that I've been talking to you guys, your eyes have been saccading back and forth between me and the frame, hopefully. I'm turning my head, you guys are fidgeting in your chairs, so you're creating your own motion as well as the motion that's being created in the world around you. Some people even argue that the world is a little bit more, you're creating more motion than actually the world around you is. At most times, especially in our world, your world is fairly static and you're moving through it. So we've already talked a little bit about screen-based reference frame. That's the motion that's in the stimulus that you're presenting. And so frame by frame, things change and move, but it's all right there in front of you, and you can calculate motion vectors based on the change across frames. However, there's also trying to figure out how your eye is influencing the visual signal, because every time you saccade, you refoveate something new. So what we can try to do is create a retinal-based reference frame. You can take the frame, and wherever the monkey's eye position landed at a given time, you leave that as the center of the world, and you move the frame to represent the space. So now it's always the frame that moves, but your eye is essentially being treated as if it was static. And you can do this over and over and over again, and this can mimic something like a retinal reference frame. And so this is just a way to kind of understand what we mean by this type of thing. And so your eye is constantly creating all this motion, which would be calculated by doing any kind of motion algorithm on successive frames in this larger field of view. So we did something similar to that. What we did was we took the trial by trial variability and we ran a regression looking at how the stimulus motion, which I've already showed you, activates the brain. Here you can see that same motion map from earlier. It's activating a lot of the superior temporal sulcus, V4 into the lateral temporal lobe. And we can look at the activity caused by eye movements by looking at changes in the velocity of the eye movement over time on successive trials. And so what we see is a very different map of activity when we use eye velocity as compared to stimulus velocity as the regressor on the brain. And it's primarily in early visual cortex and down into the ventral aspects of like V4 and stuff like that. A more clear view of this we can see by taking a difference score between the video motion versus the eye movements. We see that two distinct areas are really being driven by these two types of motion. The early visual cortex is being driven more by the retinal motion or reafferent signal, and the later visual areas like MT and on are being driven more by the stimulus motion. So what this suggests is that these later areas are accounting for this self-induced motion by subtracting it out of the signal, essentially. They're saying, I know that I caused that, so I'm going to remove it from my calculations. And then from that point on, in the rest of, for the most part, the rest of the brain, everything's just caring about what's going on in the world around you. So this is evidence that there's strong, that maybe the brain is using efference copy or corollary discharge to account for the reafference motion that's coming in through your retina at all times. Another use of these types of naturalistic stimuli is that you can start looking at how the brain is processing temporal information. And what I mean by that is coherent temporal information. So if you have more coherent temporal information, meaning a longer set of frames that are all showing you the same set of the same scene versus a shortcuts. And so Uri Hassan and his group used a method where they scrambled a set of movies at different time frames and then looked at how well correlated subjects were to both themselves and other subjects based on these different scrambling lanes. And what they found that was very interesting is that as you go from the posterior portions of the brain, the occipital lobe, to more anterior areas, the amount of temporal information that's necessary, or the temporal receptive field of the region, gets larger. So later areas like the FEF, which is making decisions about where to look and things like that, it has a much longer time scale. It's integrating information over 36 seconds in their study. And if you looked at, but it was not correlated with itself if you were looking at shorter things like 12 seconds or 4 seconds. Whereas the visual cortex, it didn't care. It didn't care how long of a time frame you were using. It just kept responding similarly if you showed the same piece of information. And so this can go a long way towards trying to understand how the brain is taking in semantic information. Because as I was saying earlier with auditory and scenes like social information, we need a set of time to actually understand how something is occurring. You can get a lot from a still frame. Maybe if you see someone winding up to punch in a still frame, you can get a sense that they're aggravated, but you might not know about what. And there's a lot more subtle cues that take a bit of time to understand what's going on. And these types of analyses with these movies can allow you to start understanding these longer scale responses. Similarly, we can start looking at other types of content. So what we did was we presented a set of movies that had social content versus movies that had no social content whatsoever. And what you're seeing here is the overall correlation to both sets of movies individually. And now, what you're probably thinking is those look remarkably similar, regardless of whether or not there's social information or not. And that is very true. They actually are quite similar. But what we can then do is compare the responses to the two types of movies, and we can get a difference map based on those. And you can see that there actually ends up being one major hotspot that's actually showing a difference between the social and the non-social movies. And we can blow that up and look at that on a flat map, and what we see is it actually overlaps with separately coded for set of face patches in the monkey. In particular, it's with a subportion of the middle lateral and middle fundus face patches. Those particular ones aren't of any importance to our talk today, but it's more that what we see here is by using these more naturalistic stimuli, we actually see that the majority of the areas that are known to be processing social information are actually responding quite similar to each other when we use a more naturalistic stimuli, suggesting that maybe these areas are actually doing something a little more complex, and when we only ask them face versus house, they go, screw it, face. But that might not be the actual coding in that area. There might be something much more complex than just whether or not it's a face or not. Obviously, a lot more research is necessary to determine those things, but I think this is strong evidence that when you start using more complex stimuli, simple responses don't always work out as well as we'd like them to. So that leads on to another set of studies that starts looking at something kind of similar to that, which is how do individuals respond similarly to the same set of stimuli. So Uri Hassan's group started a set of projects looking at this, and what they did was they presented a set of subjects with a movie, and they had them watch it twice, and then they have this response to the same subject and responses for across 2 viewings and different subjects across those same set of viewings. And what we see is that within subject, there's extremely high correlation across viewings. Between subjects, there's still a relatively high correlation. So subjects are actually viewing movies quite similarly, but it's quite reduced from what an individual subject is doing. So there's variability across subjects. What's most interesting, and I brought this up earlier, is that using different types of movies really changes the correlation across subjects in watching these movies. So they had started with The Good, the Bad, and the Ugly for no particular reason, I believe, at the first, and it actually does an extremely good job of driving the individual subject's brain similarly. The one that did the best is actually an Alfred Hitchcock movie. But then as you start going to these the Charlie Chaplin movie and Washington Square Park, you see that the correlation between subjects actually goes down quite a bit. And so the different content and different way that these films are cut and shown has a extreme influence on how individual subjects are actually processing these stimuli. So one has to be careful about simply just choosing one stimulus and going with it in these types of more naturalistic settings. You need to really vary your stimuli to make sure that what you're capturing is actually something to do with generalized real world as opposed to simply the response to that particular stimulus you chose. We can use these types of techniques, these cross-subjects techniques, to also start to try to understand cross-species brain activity. And so we can take the activity of, so this is Mantini and Van Doffel ran a study looking at the responses between humans and rhesus macaques watching the same set of movies. This was in collaboration with Uri Hassan's group, so it actually also uses The Good, Bad, and the Ugly as their stimulus. The problem here, and this is one that we're trying to work out as well, is it's hard to have a good stimulus that's species specific for both humans and monkeys, or if you want to include rodents or other types of things. Each group is going to have their own species-specific movie, and you have to kind of make a trade-off on what's going to be the best one to use. Since they knew the responses to The Good, the Bad, the Ugly worked well for humans, they started with that. We're running a similar project currently looking at 3 different species using the macaque movies, so that's going to have its own trade-offs between the species. But what they can do is they can take an ROI in humans and a set of ROIs in the monkeys and look at the responses both within the same species and across species. So you can take the ROI here and try to map it to the responses in the other species. And you do this on a ROI or voxel by voxel basis. So you don't just have to find the one voxel that's responding best to it, but you can actually look and see the full brain activity in the other species to the one voxel in, I guess it's not response, but correlation to the other species. And you can look at what areas load most across species on these things and how well do they match up. And what they found is a very similar set of maps across species watching these 2 videos. And that when you look at particular subregions, that sometimes the area matches up very well across species, but other regions actually move in space across the brain between a rhesus and a human. And while this is no proof, it helps to suggest functional homologies across species. And this type of analysis can be used to try to better understand where either, say, a monkey person should probe to look at a given feature response that they've seen in humans. If they can get them to watch the same set of movies, they can find the functional homology and go there and record. Or vice versa. In monkeys, if you compare it to humans, you may get a better sense of how you think the results of one area would apply to human data, given that the homology may not be the straight functional blow-up of the brain. So I already mentioned that we're starting to do this between other species. So what you see here is the correlation of a macaque brain to itself watching a set of movies. Across 15 different viewings, you can see that they're individual voxels are extremely well correlated with themselves. A1 here is actually a very nice control analysis here in the sense that there was actually no auditory stimuli in our movies. We had cut those out. And basically A1 is not responding during the movie presentation, and there's zero correlation there. So what we can do then is we can calculate the independent components we see in the macaque brain, and we get a spatial component, and these voxels load particularly well on independent component 1. And those are correlated with certain features in the brain, not very well with motion, but highly correlated with the contrast feature, the time course for this independent component. And then we can run the same analysis on a set of marmosets that watch the same movie. And what we can see is that across the two species, the independent components are quite similar for a number of different features. So this is a feature that's correlated with contrast. Both of them are activating early visual cortex. The motion feature, the one that's correlated with motion, is activating MT in both species. And a component that's loading on faces is activating the face patches in both species. But there's also differences across the loadings, and so we're not quite there yet, but you can start to probe what these differences mean in terms of any change in the brains between these old world monkeys and new world monkeys. Finally, we can start using these naturalistic stimuli to probe across methodologies. This works particularly well when you're working with animals. So we can have a monkey in a scanner, and we can record fMRI activity in addition to single unit recordings in a given region. Using the same stimulus, though, we can entrain both the neurons and the whole brain to the same visual space, and then try to understand how these two areas, this small scale versus large scale, are interacting with each other. So we have a set of microwire arrays in the monkey's brain. We record the cell response to I think this is 10 viewings of the same movie. You can see here that there's bands of activity at certain time points. The cell is well correlated with itself. And then we can look at the response of how that cell its response is similar to the voxel time courses when the monkey was watching the movie in the scanner. So this particular cell, it matches very well with the face patches, and this makes sense because this cell came from a face patch. However, what's interesting is all of the electrodes that we recorded from were in a face patch, and not all of them correlate with the brain in the same way. So even though these cells are within 500 microns of each other, well within the same voxel, they're actually showing extremely different patterns of response between themselves and across the whole brain, how they're correlated with the whole brain activity at the fMRI level. And so we think we can use this type of differential response across scales to try to understand connectivity is not the right word, but functional networks in the brain connected to a particular area. So that's the whole talk. Just to summarize, one thing to take away is that there is a large space in which you can place your experiment, and each choice you make doing that is going to involve a lot of trade-offs. And those trade-offs are really critical to understand when both designing and trying to analyze your experiment. We believe that using these naturalistic stimuli can be a strong step towards understanding how the brain processes the world, and it just takes a little bit of work to try and understand these features. So I showed you a bunch of different ways we can do that. One is through something like a feature regression, using stimulus and behavioral comparisons, so we can understand how the behavior is involved in processing these stimuli as well. Temporal responses across the brain are critical when dealing with these naturalistic stimuli. And the actual content of the movies can really change the way the responses are being shown. Similarly, it gives interesting outlook on intersubject ratings and interspecies similarities, and possibly between methods as well. So I want to thank you guys for listening to my talk, and I'd like to thank our lab and collaborators, and these are some of the references I mentioned during the talk. Thank you. If you have any questions, I'm happy to take them. If you could use the mic, the people recording would be very grateful. Great, I did an awesome job. Thanks for coming, guys.